Hello everybody! Today we're going to be going over central dogma. Let's get started. Central dogma states that the genetic information flows in one direction, essentially creating proteins from DNA. So in both prokaryotes and eukaryotes, DNA provides the information needed so that the cell can perform all of its functions. To do this, the DNA is transcribed into an mRNA molecule. Then the mRNA provides code to form protein, and these two processes are known as transcription and translation. So through the process of transcription and translation, a protein is built for specific sequences of amino acids that was originally encoded in the DNA. Central dogma states that DNA encodes RNA, and RNA encodes proteins. What this means is that the genes from the DNA specify the sequences of the mRNA, which in turn specify the sequences of the proteins. So now the copying of DNA to mRNA is relatively straightforward, with one nucleotide being added to the mRNA strand for every complementary nucleotide read in the DNA strand. However, the translation to protein is a bit more complex because groups of 3 nucleotides can correspond to one amino acid of the protein sequence. So here we have transcription, and down here is for translation. So now let's look at the exact process for transcription. Both eukaryotes and prokaryotes perform the same fundamental process of transcription. The difference being that for eukaryotes, transcription occurs in the nucleus, and the mRNA must be transported to the cytoplasm. Whereas in prokaryotes, the transcription just occurs in the cytoplasm. Also, for both prokaryotes and eukaryotes, transcription occurs in 3 main stages. The first stage being initiation, second being elongation, and finally termination. Transcription always proceeds from one of the 2 DNA strands. It occurs in the template strand. The mRNA product is complementary to the template strand and is almost identical to the other DNA strand called the non-template strand. They're almost identical, except that RNA contains uracil, so in place of thymine found in DNA, RNA contains uracil. During elongation, an enzyme called RNA polymerase proceeds adding nucleotides by base pairings with the DNA template. Now let's move on to translation. The synthesis of proteins is one of the cell's most energy-consuming metabolic processes. The process of translation involves decoding mRNA into a polypeptide product. And in addition to mRNA templates, many other molecules aid in the process of translation. There are, of course, mRNA templates, but there are also ribosomes, tRNAs, and different enzymic factors. For prokaryotes, the ribosomes are located in the cytoplasm. And for eukaryotes, ribosomes are in the cytoplasm and also the endoplasmic reticulum. Ribosomes are made up of 2 subunits, one smaller subunit and one larger subunit. The smaller subunit is responsible for binding mRNA templates, and the larger one is responsible for binding tRNA. Here we have the codon table, or you can call it the genetic code table. It's essentially the complete set of relationships among amino acids and codon. Each amino acid is defined by a trinucleotide sequence called a triplet codon. Translation of mRNA converts the nucleotide-based genetic information into a protein product. And the relationship between the nucleotide codon and its corresponding amino acid is called a genetic code. I know this may seem like a lot of information, so feel free to pause, rewind, and even re-watch the video if needed. And that is it on our video covering the basics of central dogma. In our next video, we're going to be covering some examples and review what we've learned. So make sure to go check that out. Thank you so much for watching. Make sure to like and subscribe.